Polymerase chain reaction or PCR technique.
I am Dr. H. Ramasubbareddy, lecturer in zoology, working at SVB Government Degree College, Kovilakuntla. Today, our topic is basics of polymerase chain reaction. Polymerase chain reaction is also called simply PCR. This test is also called molecular photocopying method. This polymerase chain reaction is very essential to synthesize more copies of DNA. Now, the learning outcomes. Students shall understand about polymerase chain reaction. They shall know the invention of PCR method. Student will understand the procedure of polymerase chain reaction. They will know the different types of PCR techniques and the application of polymer chain reaction also they will understand. Next, introduction of polymerase chain reaction. The polymerase chain reaction was not a discovery rather than an invention. Carey Mullis, the inventor of PCR in 1983, was awarded Nobel Prize in Chemistry in 1993. A special DNA polymerase, that is Taq polymerase, is used to make many copies of a short length of DNA. That means defined length of the DNA will be produced in this method. Carey Banks Mullis is an American biochemist. He discovered or invented this method. Next, polymerase chain reaction is a technique which is used to amplify the number of copies of a specific region of DNA in order to produce enough DNA to be adequately tested. PCR is a enzymatic process in which a specific region of DNA is replicated over and over again to yield many copies of particular sequence. The most widely used target nucleic acid amplification method is the polymerase chain reaction. The purpose of a PCR is to make a huge number of copies of a gene. Now it now becomes possible to analyze and characterize DNA fragments. These are the PCR machines, PCR thermal cyclers. Different PCR thermal cyclers used in different methods of PCR techniques. There are so many types of PCRs. We will discuss later about the types. These are the different PCR machines. All are used in different types of PCRs. Next, mechanism of PCR. Starting with one original copy, an almost infinite number of copies can be made using PCR. Amplified fragments of DNA can be sequenced, cloned, probed, or sized using electrophoresis. Defective gene can be amplified to diagnose any number of illness. On the screen, you can observe the sequence of isolation or the sequence of isolation n number of DNA copies you can observe on the screen. This is the PCR machine. After completing uh, production of more number of DNA copies, they are be made into agarose gel electrophoresis. These DNAs are placed in electrophoretic apparatus, and we can isolate the large and small length of DNA fragments. By using UV transilluminator, we can observe the bands on the X-ray photography. Next, what are the reagents required for PCR? These are the reagents: 1x buffer and magnesium chloride and different types of DNTPs and primers, DNA polymerase enzyme. This is also called a Taq polymerase. This special Taq polymerase is extracted from the thermobacteria, the bacteria which are living in the hot springs. From that bacteria, this Taq polymerase extracted. This Taq polymerase is essential to produce more number of DNA copies. Next, DNA sample copy. These are the reagents, chemicals required for PCR technique. What are the components in PCR? There are different components in PCR, but these are assembled in a PCR machine to perform the function. First, helicase enzyme is required to unwind the DNA fragment, and heating also required to melt the DNA fragments. Single-stranded binding proteins are also required to separate the DNA fragments. DNA polymerase, that means Taq polymerase, is essential for polymerizing the DNA. Primase is enzyme is required to providing a primer base. Primer should be added to the reaction mixture. The ligase enzyme also required to joining the fragments of DNAs. All these enzymes, proteins are placed in a PCR machine to perform the function. These are the components essential in PCR machine. Next, thermal cycling. What is the process? The process is very liberal, very step by step we will understand. A PCR machine controls temperature. Typical PCR go through the steps. There are three or more sometimes, and the three steps are essential. Sometimes four steps are performed in this activity. Next, PCR machine is also called thermal cycler. The activity undergone in the PCR machine is called thermal cycling. A PCR machine controls the temperature during the process of thermal cycling. A typical PCR go through four steps. There are four steps in polymerase chain reaction. Number one is denaturation. Number two, annealing. Number three, extension. Number four, cycling. These are the four steps are essential to produce more number of copies. First step is denaturation. To convert double-strand DNA into single strand, the DNA must first be heated to high temperature up to 95 degrees centigrade for 15 seconds. Separation of each strand acts as template DNA synthesis. Usually, a thermal cycler machine is used for this, which can quickly heat and cool samples. Here you can observe the double-stranded DNA. After heating up to 15 degrees centigrade for 15 seconds, this double-stranded is converted into two single-stranded DNA. Second step is annealing. Annealing following denaturation, the temperature is decreased to 60 degrees for 60 seconds so that the primers can bind the single-strand DNA. Primers are brief synthetic DNA sequences that complement the target sequences. Here you can observe the primers which are attached to the single-stranded DNA. Next, third step, extension. Nucleotide triphosphate and a thermostable DNA polymerase are added to the reaction mixture. The temperature is increased to about 72 degrees centigrade to activate the DNA polymerase enzyme. The DNA polymerase accelerates the polymerization process of primers and extends the primer resulting synthesis of target DNA sequence. The Taq polymerase are used in PCR technology. This Taq polymerase is extracted from the thermostable bacteria, generally which lives in the hot springs which are there in the world. This Taq polymerase is essential for the PCR technique. In this picture, you can observe the extension process. By using primer, the process, the polymerization process is carried out from 3 end to 5 end. This is the extension process. Next, fourth step is cycling. Depending on the concentration of starting DNA and the required level of amplification, step 1 to 3 are repeated for a predetermined number of cycles, often 20 to 40 times. The amount of DNA doubles with each cycle, increasing the target sequence exponentially. In this diagram, you can observe first the template DNA. In first cycle, this template DNA can produce 2 to 4 copies. These 4 copies in second cycle gives 8 copies. The 8 copies in third cycle gives 16 copies. In fourth cycle, it produces 32 copies. Likewise, the DNA amplification takes place. This is the exponential amplification of template DNA. This is the total reaction. PCR polymerase chain reaction, you can observe the total steps in this diagram. This is the double-stranded DNA. In the first step, denaturation, double-stranded DNA can convert into two single strands. In the second step, annealing. During the annealing, a primer is added to the single strand of DNA. In the third step, extension. Basing on the primer, the DNTPs are added to the primer to extend the DNA fragment. This is the total diagram of PCR technique. There are different types of PCR machines. There are different types of PCR techniques we can observe. There are more than 20 to 25 types of PCR techniques are there for different purposes. First one is RT-PCR. This is also called reverse transcription PCR. Second one is colony PCR. Third one is standard PCR. Fourth one is nested PCR. Fifth one is digital PCR. Sixth one multiplex PCR. Seventh real-time PCR. Eighth inverse PCR. Ninth long-range PCR. 10th ligation mediated PCR. Likewise, there are different types of PCR for different purposes. Next, applications of PCR. PCR is an extremely simple yet immensely powerful technique. It allows enormous amplification of any specific sequence of DNA provided that short sequences either side of it are known. Allow faster diagnosis and identification while enhancing sensitivity and maintaining specificity. There are number of applications for this PCR techniques. During DNA sequencing, this PCR technique is essential to sequence the DNA strand. In genetic disease diagnosis, testing of genetic diseases, disease mutations, and monitoring the gene in gene therapy. This technique is also useful in detecting disease causing genes in the parents. In the forensic analysis, this PCR technique is used as a tool in genetic fingerprinting, identifying the criminal from millions of people also it is useful. It is useful to identify the baby, which parents of the baby. That means paternity testing. PCR also useful in paternity testing. In the microbial detection, PCR technique is used to detect the microbes. They can analyze the DNA sequence by using the sequencing technique with the help of PCR. PCR technique is also useful to monitor the environmental conditions. PCR techniques is also useful in research. Scientists can compare the genome of two organisms in genomic studies. In the phylogenetic analysis of DNA from any source such as fossils. This technique is also useful to analyze the gene expression. This technique is also useful to diagnose corona diagnosis. We all are experienced the pandemic time of corona. This corona virus is tested by using the RT-PCR technique. These are the applications of PCR. Next, these are the references to further study of PCR technique. Next, thank you for all.
Please subscribe Ramos Zoology.